Welcome to the Drought and Evapotranspiration Lab activity. At this point, you should have already listened to the Drought and Evapotranspiration Lab PowerPoint. If you haven't, go back and do that first. This lab activity follows an experiment. Using peppers, we've created treatments to reflect water stress conditions and well-watered conditions to see differences in plant productivity. Let's start by forming our research question. Normally, you want a very focused question so that you can clearly answer it. In this case, we're trying to keep it refined, but it's still a little broad. For the purposes of this class, this is okay. We'll be observing effects of drought stress over the course of 4 weeks, and we want to know the impact this will have on plant biomass and other plant features. We're going to make general observations, perform stem height and leaf count measurements, and then measure plant dry weights. Our control treatment is going to be a set of pepper plants that we watered before there was any sign of wilting. This means we were replacing the water lost from evapotranspiration. We're calling it 100% ET. Our other treatment is a set of pepper plants that will only be watered after we notice signs of wilting to create some conditions of water stress within the plants. Although we won't necessarily make this measurement, it is possible to measure ET directly by weighing the pot. The loss of weight over time represents water loss through evaporation and transpiration. We're going to make some general observations before the end of the experiment and note color, size, and overall visual differences. But after the 4 weeks, we have a couple things to record. One of them you're already familiar with is plant height, and we're going to record leaf number 2. Leaves are source organs because they are the site of photosynthesis. Drought stress impacts leaves and inhibits photosynthesis, so it will be interesting to see any difference in leaf number after the 4 weeks. Root and shoot growth reflects resource availability, and we need to measure root and shoot fresh weight to compare plant size. You might think this fresh weight measurement alone can tell us about plant productivity. Heavier roots and shoots are probably bigger and contain more assimilates and other biological compounds. Unfortunately, fresh weight is just too variable and easily skewed. Even after removing them from soil and cleaning them, roots can still pick up moisture, and so fresh weight is not an accurate reflection of plant biomass. Because of this, it is more common to measure plant dry weight. With water weight removed, dry weight is a much better measure of resource and photoassimilate production and allocation.